Let's talk about the maturation factors which are required for the erythropoiesis. As I mentioned earlier, there are two vital maturation factors which are required for the formation of the RBCs. Now, these vital maturation factors is one, the folic acid, and second, the vitamin B12. Now, both of these are taken through the diet. The daily recommended allowance for the folic acid is 0.05 milligram per day, while for vitamin B12 is 0.001 milligram per day. Now, this amount on a daily basis has to be met for the optimum functionality of the erythropoiesis. Now, both of these, both the folic acid and vitamin B12, when taken through the diet, they pass through the oral cavity and into the stomach. Now, when they enter the stomach, another thing that happens is that stomach releases the intrinsic factor. Now, intrinsic factor is a protein which is released by the cells of the stomach. And without the intrinsic factor, vitamin B12 cannot be absorbed through the intestines and then go inside the parenchyma of the liver. So, when they are inside the stomach, with the help of intrinsic factor from the cells of the stomach, vitamin B12 is absorbed into the portal circulation, while the folic acid is simply absorbed into the portal circulation and then to the tissue of the liver. And this happens through the stomach, then to the lumen of the intestines, and then to the tissue of the liver. Now, when they are present inside the tissue of the liver or the parenchymal cells, they are present in their active forms. Vitamin B12 is present in the active form as methylcobalamin, while folic acid is present in its active form as the tetrahydrofolate. Now, this N5 tetrahydrofolate can be further converted into its more active form, that is the tetrahydrofolate. And the conversion of the N5 tetrahydrofolate into the tetrahydrofolate is facilitated by the methylcobalamin. Now, when methylcobalamin converts the N5 tetrahydrofolate into the tetrahydrofolate, only then is the folic acid is able to take part in the folic acid cycle. Now, once tetrahydrofolate has formed, it takes part in the folic acid cycle. And this is the folic acid cycle where there is a continuous usage and then again reappearance of the folic acid. It converts from the tetrahydrofolic acid from the dihydrofolic acid. So there is a continuous usage and renewal of the folic acid in the folate regeneration cycle. And because of this folic acid cycle, what happens is that there is a conversion of the deoxyuridylate into the deoxythymidylate, or simply put, the uridine is converted into thymidine. Now, we both know that thymine is one of the important components or one of the building blocks of the nucleic acids, and deoxythymidylate is one of the primary building block of the DNA. So, because of the folic acid and the folic acid regeneration, the uridine is converted into the thymidine. And because of this, again, the DNA is formed, the DNA is expressed, and because of the expression of the DNA inside of the RBC is proper, the RBC matures properly from a reticulocyte to a biconcave erythrocyte. So this cycle, when happens, can give the deoxythymidylate to the sites of the erythropoiesis. Now, when this building block of nucleic acid comes to the sites of the erythropoiesis, it takes part in the formation of from the non-committed progenitors to the committed progenitors, then all the way to the erythroid progenitors into the full-fledged erythrocyte. And then that erythrocyte is when passes through into the bloodstream, is converted into a full-fledged biconcave anucleated RBC or erythrocyte.